Hey there, and welcome to This Explainer. Today, we're diving headfirst into literally one of the most fascinating mechanics of human biology, the nervous system. We're going to decode the very sparks that make you, well, you. It's a journey into the incredibly elegant microscopic engineering that keeps us alive, thinking, and moving every single day.
I mean, think about this grand communication challenge for a second, right? How does our massive physical body rapidly coordinate and integrate all of its activities? When you touch a hot stove, how does your hand know to yank itself away before you've even consciously registered the pain? It all happens in fractions of a millisecond. When you frame the nervous system this way, it really reveals itself as an absolute biological engineering marvel.
So here's our roadmap for today. We're going to kick things off with our body's command center, then look at the anatomy of a neuron, dive into the resting potential, the action potential, how we reset the system, and finally, how we go from a signal to a sensation.
All right, Section 1, our body's command center.
Our biological hardware is brilliantly split into two main parts. You've got the central nervous system, or CNS, which is your brain and spinal cord. Think of this as the ultimate integration and interpretation center. But it relies entirely on the peripheral nervous system, or PNS. The PNS is out there doing the legwork, bringing all that vital sensory information in from the outside environment. It's the ultimate two-way street of data.
Moving right along to Section 2, anatomy of a neuron. Now, a neuron doesn't really look like your standard textbook round cell. It is totally built for rapid communication. Signals are received at one end by those branch-like structures called dendrites, and they filter right down into the cell body. From there, if the signal is strong enough, it travels down that long tail called the axon. And notice how the axon is wrapped in a myelin sheath. That acts perfectly as biological insulation, letting the signal travel super efficiently all the way down to the synaptic terminals.
And at the very end of that incredible journey, down at the synaptic terminal, that electrical signal actually translates into a chemical one. The neuron releases tiny neurotransmitters across a microscopic gap called the synaptic cleft to bind with receptors on the next cell. This is exactly how neurons pass the message along and communicate with other excitable tissues.
Which brings us to Section 3, the resting potential.
Okay, so think of a cell at rest like a tightly coiled spring, just absolutely packed with potential energy. To understand this, we have to grasp two distinct forces at play across the cell membrane. First up is the electrical gradient, which is simply the difference in the overall net charge between the inside and the outside of the cell. Second is the chemical gradient, and that's entirely about the concentration of specific ions, namely sodium and potassium.
So, the absolutely crucial number to remember here is negative 70 millivolts. This is the standard resting membrane potential. It sits right at negative 70 because there is a lot more positive charge on the outside of the cell compared to the inside. Specifically, we've got a whole bunch of sodium crowded on the outside and potassium hanging out on the inside.
Now, I want you to actively think about this for a second. The cell uses active transport channels called sodium-potassium pumps that constantly burn energy to maintain this exact ratio of ions. What do you think would happen to that resting membrane potential if you were to suddenly just stop that pump?
Well, the answer is that the membrane potential would totally lose its negative charge. Without the active transport of the sodium-potassium pump constantly pushing ions against their natural gradients, the ions would just leak across the membrane until they reached equilibrium. That pump is quite literally the engine that keeps our biological battery charged and ready to fire at a moment's notice.
All right, Section 4, firing the action potential.
Remember how we just said we start at negative 70? Well, negative 55 millivolts is the magic threshold. If a stimulus causes the inside of the cell to become just positive enough to hit negative 55, it crosses the definitive point of no return. Boom! An action potential is officially triggered.
Imagine a giant row of dominoes. Action potentials are literally all or nothing. You don't get a half spark or a weak spark. Once you tip that first domino, the signal travels down the axon through what we call non-decremental conduction. Basically, that's just a fancy way of saying it travels long distances through the neuron without ever fading or losing strength.
Let's break down this spark, and I mean rapid. This whole thing takes just 0.001 seconds, strictly by its four distinct phases. Step 1 is depolarization. Sodium channels open, and positive sodium rushes in. Step 2 is the peak. The sodium channels shut down, and the potassium channels open up. Step 3 is repolarization. Positive potassium rushes out of the cell to lower the charge.
And finally, step 4 is hyperpolarization, where the cell actually overshoots its resting state slightly before finally settling back down.
If we trace this massive electrical spike on a graph, it's a pretty dramatic journey. You can see the triggering event pushing the voltage up from -70. Once it hits that dash threshold line at -55, it absolutely skyrockets up into positive voltage territory as the sodium floods in, hits its peak, and then plunges right back down during repolarization.
Now let's connect that voltage spike to the physical reality of the cell membrane. Because these protein gates opening and closing aren't just random. They are voltage-gated. They are precision mechanical doors triggered by exact millivolt changes. The rapid opening of the sodium channel right at -55 millivolts is exactly what causes that massive upward spike on the graph we just looked at.
That takes us to section 5, resetting the system.
So how does the neuron recover? It uses refractory periods. During and immediately after an action potential, the neuron enters a state where it is either very difficult or utterly impossible to trigger another spark. And this is brilliant biological design. It limits our maximum transmission rate so signals don't overlap into total chaos, and it ensures the action potential only ever flows forward in one direction down the axon.
And finally, section 6, from signal to sensation.
We've spent a lot of time zoomed in on the microscopic details, so let's zoom back out for a minute. When sensory information from your environment triggers these action potentials, the afferent division carries those signals inward. But sometimes they don't even go all the way up to your brain first. Sensory neurons can communicate directly with your spinal cord to trigger a rapid reflex. It's a really primitive form of protection. Fun fact, human babies are actually born with a ton of these automatic reflexes that simply disappear as their brains grow and develop higher processing power.
But for the signals that actually do reach your brain, how much processing power do they get? Well, it all depends on the receptive field. If a part of your body has a small receptive field with a ton of densely packed receptors, like your fingertips, for example, your brain dedicates a massive portion of the sensory cortex just to process it. On the flip side, areas with few receptors covering a huge area get way less brain real estate.
I want to leave you with a really thought-provoking question to chew on. We established earlier that action potentials are an all-or-nothing domino effect, right? They don't change in size. So if every single action potential traveling up your arm is just an identical, uniform spark jumping from -70 to +30 millivolts, how does your brain possibly know the difference between a gentle, loving touch and a sharp, sudden pain? Think about that incredible translation happening in your mind right now. Thank you so much for joining me for this explainer. Keep wondering, and I'll see you next time.